Good afternoon and welcome to everyone. Let me wish you also a happy new year. It is a pleasure for me to present a new seminar from Showcasing Discoveries at Josep Carreras Research Institute, and I'm very happy to present you the group leader of cancer genetics, Dr. Montse Sanchez-Cespedes. She's a biologist and obtained her PhD from the Universitat de Barcelona in Spain. She was a postdoctoral fellow at the Johns Hopkins University School of Medicine at USA, and there she focused on the identification of novel genetic and molecular alterations in lung cancer. Among her main scientific contributions during this period was the discovery that LKB1 was genetically inactive in lung adenocarcinomas. Today, this gene is a well-established tumor suppressor gene involved in lung cancer development. From 2001 to 2008, she was the group leader of the lung cancer group at the Spanish National Cancer Center in CENIO in Madrid. And then she moved for 1 year to Bellvitge Biomedical Research Institute, IDB, in Barcelona. Currently, she leads the group Cancer Genetics at the Josep Carreras Research Institute here in Badalona. And her laboratory is devoted to the genetic, epigenetic, and molecular study of the mechanisms that drive cancer development, especially focusing in lung cancer. Her team uses the latest high-throughput sequencing technologies to create profiles of the recurrently altered genes in cancer. They are also interested in understanding the mechanism by which the abnormal function of these genes contribute to cancer development. The purpose of her research is to implement the clinical management on cancer patients and to design novel therapeutic strategies. Dr. Sanchez-Cespedes is a member of international scientific societies, editorial board member of the Journal of Pathology, and reviewer for many journals and funding agencies. Let me say that she has an age index of 50 thanks to over 100 original publications and reviews in international and peer-reviewed journals of prestige. Before starting the presentation, just remind you that you can ask questions using the question mark button, and it will be a pleasure to listen to you, Dr. Sanchez-Cespedes.
Thank you very much, Marta, for your kind introduction. So today, in this presentation, I will be talking about the determinants of immunological evasion and the immunocheckpoint inhibition response under a genetic point of view. As Marta just said, we are the cancer genetic group at the Josep Carreras Research Institute, and our main goal is to contribute to the catalog of genes that are mutated in lung cancer. We want to understand their role in cancer development, and the ultimate goal of our work is to use this knowledge to implement cancer therapeutics. Currently, we have 3 main projects ongoing in the lab. One of them is about the subject of immunoscape and immunotherapeutics that I will be talking to you today. Another one is the understanding of the genetics, the genetic alterations that underlie or that drive the genetic abnormalities in cancer development. In this regard, we have been working with the Swiss chromatin remodeling complex, also with the MYC and MAX transcription factors, and also more recently with the non-canonical PRC1 complex, dependent on MEGA, among others. Another important subject that we work in the lab is the identification of novel targets for therapeutics, and also in the understanding on which are the mechanisms that drive acquired resistance to these targeted therapeutics. Lung cancer is the first cause of death due to cancer, despite being the 3rd in frequency. And this bad prognosis is mainly because of 2 reasons. One is the late diagnosis. 80% of the lung tumors are diagnosed at late stages. And another one is the low response to the traditional therapeutics. The therapeutics in lung cancer had barely changed until the year 2004, in which the first targeted therapy were developed for this type of cancer. These therapeutics were inhibitors of the tyrosine kinase activity of some growth factors, such as EGFR, ALK, ROS, RET, etc., among others. The problem is of these therapeutics. There are several problems, but the main problem is that only a few subset of patients can be treated with these therapeutics. Only those whose tumors carry genetic activation at these receptors, and these are less than 5% of all the lung cancer patients. More recently, the immunotherapeutics have started to be administered to lung cancer patients, and this has been really a revolution in the treatment of this disease. About 50% of advanced lung tumors are currently treated in one way or another with immunocheckpoint inhibitors. However, the main problem, or one of the main problems, is that despite this is widely used, we still don't have very good markers that determine which patient will truly benefit from these therapeutics and which one will not. And this is despite we have wide and very extent knowledge about the genetic of the lung tumors, thanks to the development of the new new sequencing technologies. The relationship between the immune system and cancer development has been known for a long time. There are two different sides that tell us about the importance of the immune system in recognizing and in killing cancer cells. One is the fact that the different types of immunodeficiency, for example, AIDS, the administration of immunosuppressor, or several immunosuppressor-related diseases, all these conditions have an increased risk for developing cancer. On the other hand, several years ago, during the last years, many years ago, it had been discovered very spontaneous and anecdotal cases, but still very interesting, of patients whose tumors have regressed completely after a very acute infection. And these observations were the basis for the first vaccines against cancer. In most solid tumors, especially in lung cancer, the therapeutics, the immuno-related therapeutics, are monoclonal antibodies that target PD-L1 or PD-1 molecules. These molecules, what they do is to inhibit the immunoresponse. In a normal immunoreaction, the tumor cell or the antigen-presenting cell present the antigens to the T cell, and these recognitions triggers an immunoreaction. However, at some point, these immunoreactions needs to be suppressed in order to prevent an overactivation of the immune system that can be detrimental for the organism. And as I say, this inhibition takes place through the interaction of PD-1 in the T cell and PD-1 in the tumor cell or the antigen-presenting cell. These immunotherapeutics are immunocheckpoint inhibitors that are monoclonal antibodies that bind either PD-1 or PD-L1, therefore inhibiting the inhibitors of the immunoresponse. In lung cancer, there have been many clinical trials that have tested the efficacy of different PD-L1 or anti-PD-L1 antibodies. Most of them have evidenced improvement in overall survival or other clinical parameters in the case of immunotherapeutics as compared with the traditional therapeutics. Perhaps the most impressive results were obtained in the KEYNOTE-025, in which they perform another clinical trial selecting the patients whose tumors have the levels of PD-L1 above 50%. When they select this type of patients, and these patients were treated with anti-PD-L1 antibodies, they show that the overall survival of those patients were double or even more as compared to the patients that were treated with the traditional therapeutics, which clearly evidenced that immunotherapeutics have a very important use in the treatment of lung cancer. Several years ago, as I said at the beginning, our group is interested in identifying alterations, novel alterations in cancer, using lung cancer as a model. And several years ago, we performed a wide genome screening, in particular whole exome and RNA sequencing, of lung tumors that have been implanted in mice. We did that because we wanted to obtain highly pure tumors, pures that didn't have any normal contaminant cells, in order to improve or to increase the sensitivity of our sequencing approach. We were able to obtain 14 different PDX, mostly lung squamous cell carcinoma and lung adenocarcinomas. The sequencing approach, this is the sequencing approach, and this is the results of some selected genes by RNA-seq. So this analysis allows us to identify mutations at well-established genes. These mutations were present at a very high frequency, even higher than you would expect if you sequence lung primary tumors, supporting the fact or indicating that we have truly increased the sensitivity of our sequencing approach. We also obtain candidate novel oncogenes or tumor suppressor genes, some of them involved in the epigenetic regulation, in cell addition, cytoskeleton, etc. But what got our attention was the observation of mutations in two genes that are involved in the immunorecognition. That is the B2M and the HLA-1 protein. These two proteins are part of the HLA-1 complex that presents the antigen to the T cell. One of the mutations was one homozygous deletion in HLA, and in the case of B2M, this was an stone mutation in the gene. We decided to study further B2M because this is a quite small gene, and the genetics are quite simple and it's not very heterogeneous, as opposed to HLA, which is a molecule that is quite difficult to study under a genetic point of view because it's genetically very heterogeneous. So we, as I said, we decided to study B2M further. Here is the representation of the HLA-1 and HLA-2 complex. B2M is a small protein that anchors the complex to the cell membrane, and the HLA, which can be either HLA-A, HLA-B, or HLA-C, is the other component of the complex. So we screen for mutations at B2M in a panel of about 200 different lung primary tumors and cancer cell lines, and we obtain mutations in about 7% of the tumors tested. Most of these mutations were mutations that predicted for a truncated protein. Stone mutations or friendship insertions of deletions were the most frequent. We observed also 3 different missense mutations. All the mutations that predicted for a truncated protein were verified to render or to not have any protein expression at all, as you can see here in this Western blot from these 4 different cell lines that had inactivated mutations. And in the case of the primary tumors, we also verified that these mutations were homozygous using microsatellite analysis and to determine the loss of heterozygosity of the normal allele. Therefore, all these observations clearly state or indicated that B2M acts as a tumor suppressor gene. We next search in databases to know whether this is a widespread phenomena in cancer, and we observe that this is the CBioPortal database, and we observe that from 1 to 5% of all human tumors carry inactivation in B2M. And as you can see here, most of the mutations, the large majority of the mutations are of the truncating type. So further supporting that B2M is a tumor suppressor gene in general in cancer. As I just showed you, most of the mutations that we got in our screening were predicted for truncated proteins. However, 3 of them were missense mutations. 2 of these missense mutations affect quite conserved amino acids, and another one affected an amino acid that is not conserved at all. We decided to study a little bit the possible effects of these mutations in the function of the complex, in particular in the appropriate positioning of the complex in the cell surface. We select different lung cancer cell lines that were deficient for V2M, and we express the wild-type version of V2M and also three of the three missense mutations. We perform immunofluorescence of V2M and of HLA-1. HLA-1 is an antibody that recognizes the three HLAs, HLA-A, HLA-B, and HLA-C at the same time. And as you can see here, of course, when we transfected the cells or we infected the cells with the anti-vector, there was no V2M in any place in the cell. However, we were able to detect HLA, but not in the cell surface. Instead, the HLA protein was stuck inside the cytoplasm. When we infected the cells with the wild-type V2M, we of course observed V2M and HLA both in the cell surface. These two missense mutations, we observed that didn't affect at all the positioning of the HLA complex in the cell membrane, indicating that these mutations does not affect the correct positioning of the complex. We don't know whether they affect other functions of the complex, but definitely not the positioning in the cell surface. However, it was quite interesting to observe that this mutation, the change of tryptophan in the AT residue, completely abrogate the capability of the complex to reach the cell surface, is supporting that these mutations somehow affect the maturation or the transportation of the complex. Here, this is the same with using another cell line that is deficient for V2M. In these cells, the observations are a little bit more difficult to be determined because these cells have very small cytoplasm, but still you can see here that when you don't have V2M, the HLA-1 proteins are stuck in the cytosol, in this case surrounding the nucleus. When you express V2M or these two missense mutations, the complex is now able, HLA and V2M, to reach the cell surface. However, with these other mutations, the HLA complex is not able to go to the cell surface. Next, we decided to determine the levels of expression of V2M and HLA-1 in a large panel of lung primary tumors. We select 274 lung squamous cell carcinomas and 145 lung adenocarcinomas. And what we observed was, we established three different categories for the immunostaining. One is negative, that is the complete absence of HLA and V2M, which usually have the same pattern of expression. Then another category was the weak category, which is the levels of HLA and V2M are somehow lower than the surrounding stroma. Here, the stroma are a very good positive control because it's always positive. So when the tumor cell had lower levels as compared to the stroma, we call this to have weak expression of these proteins. And then the strong levels of HLA and V2M, which indicates that kind of normal levels. That is the same levels in the tumors as compared to the stroma. So we observed that about 1/3 of the lung adenocarcinomas and a little bit more of the lung squamous carcinomas had complete absence of HLA complex, HLA proteins and V2M in the cell surface. Around about another 1%, 1/3 of the tumors had lower levels or weak levels of these proteins, and another third of the tumors had strong or normal levels of V2M. We next correlate the presence of complex in the cell membranes, strong or weak categories, and negative categories, with other immunorelated proteins, such as the amount of infiltrating CD8s or the levels of PD-L1, I'm sorry. And we obtained that there was a significant correlation between the higher the levels or the high levels of HLA expressing tumors and high levels of CD8 infiltrations and high levels of PDL, indicating that there are a set of tumors that tend to have kind of a positive immunophenotype. There has also been hot tumors because they are positive for most of the immunomarkers tested. And there was usually another set of tumors that tend to be negative for most of these markers. Of course, there are also some tumors that are in the intermediate stage that they have positive for some of the markers and negative for others. But in general, there was a trend to be or either positive for everything or negative for everything. We determined whether the levels of HLA or V2M had any association with overall survival of the patients. This is our stage 1 to 3 patients and treatment-naive patients. These patients have not received any treatment. And we observed that there was no correlation with the overall survival or the recurrent survival, in this case, when the patients had been treated with traditional therapeutics, not immunotherapy. When we started this study, the administration of immunotherapeutics was not as frequent. Very few patients, only clinical trials, had been treated with these novel drugs. However, we were fortunate because we were collaborating with Dr. Enriqueta Felipe at the BioBron, and she provided us with biopsies of 14 patients that had been treated with PD-L1 or PD-1 therapeutics. So we performed immunostaining of V2M, HLA, and CTL, and also PD-L1, and established a different category according to the pattern of expression, was high, moderate, or negative. And what we observed was that those patients that had either high, that were within the high or moderate categories tend to have longer survival as compared to the patients whose tumors were within the negative categories. This difference at that moment were not statistically significant, probably because the number of patients was quite low, only 14 patients. Currently, we are extending this study with a larger number of patients, and mostly patients that have been treated with these therapeutics in the first line. Now is the moment to do that because now there are really large amount of patients that are being treated with immunocheckpoint inhibitors. As I say, the HLA complex undergoes a maturation process in the endoplasmic reticulum in which several different proteins are involved. Calreticulin, TAP1, TAP2, TAPACIN, etc. All of them contribute to the maturation of the complex in the endoplasmic reticulum, and once the protein is matured, it is transported to the cell surface through the Golgi network. We reason that it could be possible that mutations or inactivation or alterations at any of these proteins might also affect the function of the HLA-1 complex. We screen in databases, in this case in the Sanger databases or in the Cancer Cell Line Encyclopedia, which provides with the genetic data for cancer cell lines, and we observe the presence of mutations in some of them. This is one example of one of the mutations that were found in TAP1 gene. This is an stop mutations homozygous supporting that these have a tumor suppressor role. However, I must say that the presence of this genetic alteration was quite low. In another project that followed the previous one, we were interested in understanding whether the immunophenotype profile, that is the immunostaining of these proteins, PD-L1, infiltrating lymphocytes, and the HLA-1 complex, were somehow correlated with the genetic status of the tumors in common or well-known tumor suppressor genes or oncogenes, both in lung adenocarcinomas and in lung squamous carcinomas. What we observed was that in lung adenocarcinomas, the presence of mutations in activation at LKB1 or STK11 is the same, tended to be associated with a cold immunophenotype or a negative immunophenotype. As opposed to that, the presence of activating mutations at the MET oncogenes tend to be associated with high levels of PD-L1. The association, these associations, unless in the case of PD-L1, were confirmed for both STK11, in which, as you can see, tumors that have mutations in STK11 tend to have lower levels of CD274, which is the name of the transcript for PD-L1. And in the case of MET, the presence of activating mutations at MET tend to associate with a higher levels of this transcript. We decided to explore these two associations a little bit further, and we started with STK11. We selected different lung cancer cell lines which are deficient for LKB1 due to inactivating mutations, and we restituted the expression of this protein. We didn't observe any changes in the levels of PD-L1 or of P2N or other markers tested upon restitution of wild-type LKB1. When we were performing this study, a paper appeared saying that those tumors that were LKB1 or STK deficient promote the recruitment of neutrophils and other pro-inflammatory cytokines that suppress the activity of the T cells in the tumor microenvironment. Because of our observations and because of these papers indicate that the activity or the action of LKB1 in the immune system is not a cell autonomous, that is, it involves the tumor microenvironment, we decided to stop the project momentarily. And we changed to understand the role of MET in the regulation of PD-L1. The MET oncogene is activated in lung cancer. About 5% of the lung tumors carry genetic activation of MET. One of the mechanisms for genetic activation of MET is gene amplification. A very high number of copies of the gene MET are found in these tumors, and this is associated with a constitutional activation of MET, which is observed by the presence of phosphorylation at the receptor. The activation of MET can also be achieved by point mutations. In this case, mutations that affect the exon intron boundaries of exon 14. These mutations are several specific mutations in hotspots. All of them led to the abrogation or the elimination of the exon 14 in the related transcript and in the translated protein. And this is because the amino acids of the region corresponding to exon 14 is the region that is the recognition sequence for the CBL protein. And this protein, what it does, it targets the MET receptor to internalize the receptor and targets it to degradation. Therefore, for a tumor cell, it's beneficial to not having this region, because in this way, the internalization and the degradation of the receptor will be more difficult, and the receptor will stay longer in the cell surface. And therefore, it will be acting as an oncogene. Here you have just some examples of some of these mutations in primary tumors and the elimination of exon 14 in those tumors that do have these activated mutations. We selected a panel of cell lines that had activation of MET, either by gene amplification or by point mutations, and also cell lines that were wild type for MET. And here, this is a Western blot that shows the levels of MET and the levels of PD-L1 in several of the cell lines selected for this study. Here I have to mention that when you do a Western blot of PD-L1, you see two different bands. The lower band, it is known to correspond to the newly transcribed protein, whether the upper band corresponds also to PD-L1, but to a protein that has suffered different post-translational modifications, mainly glycosylation. So we started with this cell line, which has an activating mutation. In the case of activating mutations, for the receptor to be active, the receptor needs to have the corresponding growth factor. In the case of MET, the growth factor is hepatocyte growth factor. So you can see that the phosphorylation of MET is enhanced upon administration of the hepatocyte growth factor. And the activation or the increase in the levels of PD-L1, as seen here in the lower band, goes in parallel with the activation of MET. Here we have used also interferon gamma, which is as a positive control because this is a well-known activator of the expression of PD-L1. Here is the same study, but using also crizotinib, which is an inhibitor of the activity of MET. And here you can see that the administration of crizotinib, here is the cells upon administration of hepatocyte growth factor, in which the increase in the PD-L1 is observed. And then when you inhibit the activity of the receptor, you see that the increase in PD-L1 is completely abrogated. And the same happened with this cell line. In this case, this cell line has an amplification and the receptor is constitutively active. But when you add crizotinib to the media, you see that there is an abrogation of the expression, the new PD-L1 protein. And here is another example. Well, in this case, this cell line is wild type for MET. Therefore, there is no MET activation. Even if you add hepatocyte growth factor to the media, you only see activation of MET when you add interferon gamma. In this particular cell line, it's very interesting, and I will come later to this case, because in this cell line, the administration of interferon gamma did not increase the levels of PD-L1. The activation of MET, it did. All the observations were confirmed to be at an mRNA level, indicating that this is a transcriptional activation, not at somehow protein stabilization protein or other mechanism. We established the MET signature using two cell lines that respond to the MET HGF administration or to the inhibition by crizotinib. And we saw that among the genes that are upregulated upon MET activations are genes that are related with the immunoresponse. And as I showed you before, interferon gamma is an activator of the expression of PD-L1. Interferon gamma is a type 2 of interferon that it binds the receptor. The receptor is an heterodimer between the interferon growth factor receptor 1 and interferon receptor 2 proteins. And this activation lets the Janus kinase proteins to bind the receptor. The Janus kinase molecules, once activated, they phosphorylate STAT1, which is now able to homodimerize and to enter the nucleus to activate transcription of different targets. Among the targets are IRF1, which is another transcription factor. There are also molecules that are involved in the HLA1 class immuno-related proteins, and PD-L1, different chemokines, etc., among others. So we decided to explore whether the activation of the expression of PD-L1 by MET was somehow related with the interferon pathway. And we activate the expression of PD-L1 by MET and by interferon and determine the levels of different molecules acting downstream of the interferon gamma pathway. And what we observed was that in this cell, remember, you administer HGF and the levels of PD-L1 are increased, and this increase was in the absence of activation of this molecule, which is a very well-known downstream target of the interferon. We also add to these cells ruxolitinib, which is an inhibitor of the interferon gamma pathway because it's an inhibitor of the Janus kinases, and you can see that this barely affects the increase of PD-L1 by HGF. However, it did really inhibit the levels of PD-L1 protein upon interferon treatment. And here you see that interferon inhibited the expression of of PD-L1 after EGF treatment. You see, as compared, there is a quite levels of protein in this case as compared to when you don't add the crizotinib. And the same with this another cell line that has gene amplification and constitutive activation of MET. These observations were also found to happen at a transcriptional level. You remember before I showed you one cell line that had activation of MET, but was not able to activate to increase the levels of PD-L1 when you administer gamma interferon. And this was a line. It was this line. So we decided to test a panel of lung cancer cell lines for their response to interferon. We were able to collect different cell lines that were not able to increase the levels of phospho-STAT or the levels of IRF1 or PD-L1 upon administration of interferon. 3 of these cell lines were cell lines that are commercially available and that the genetic status, their whole exome sequencing, is available at Sanger or the Cancer Cell Line Encyclopedia database. So we look for mutations at different molecules in the pathway of gamma interferon, and we observe that the 3 of them carry inactivating and homozygous mutations in JAK2. You can see here the mutations. These explain the lack of response to interferon. This work was the first to report JAK2 mutations in lung cancer. However, different cases of JAK2 mutations had been already reported in melanoma and to associate also with the lack of response to gamma interferon. We also use RNA-seq to determine the genetic profile of the genetic signature, gene expression signature of the response to interferon, and we obtain a signature from the 2 cell lines that respond very well to the administration of gamma interferon. However, you can see here that 2 of the cell lines that we perform RNA-seq and that were JAK2 mutant didn't show a proper response to, didn't increase the interferon gamma-related pathways upon administration of interferon, confirming that or reassuring that these cells do not have respond to this cytokine. Here I just want to mention that our observations and the observations by others in melanoma and in other cancer tumors clearly support that JAK2 is a tumor suppressor gene. However, it has been known for a long time that JAK2 is a oncogene that is activated in some myeloproliferative malignancies. This is, to me, the first case that is clearly shows that one same molecule can act as a tumor suppressor gene or as an oncogene, depending on the tumor type. In this case, the oncogenic function of JAK2 is the reason for JAK2 being an oncogene in this type of tumors is because JAK2 is a transducer of the signal of the thrombopoietin receptor and of the erythropoietin receptor, which are receptors that are involved in the cell growth of different erythroid progenitor cells. So I think this is a quite interesting case that, as I said, a molecule can act as an oncogene or tumor suppressor gene dependent on the tumor type. Because we had the gene expression signature of interferon gamma and of MET, we decided to see whether there was a common ground between the 2 signatures. And we observed that really there were very few genes that were commonly upregulated or downregulated in both signatures. But it was interesting to observe that among the genes that were commonly upregulated in both signatures were several genes that what they do is to suppress the immune response. One is PD-L1, another one is PD-L2, and SOX3 or SOX1, etc. So it is quite interesting because the administration of interferon downregulates or suppresses the immune response to prevent the overactivation of a complex. So the interferon first increases the immune response, but after a while it suppresses the immune response to avoid the detrimental overactivation of the immune response. However, in the case of MET, it could be that since it is an oncogene, it requires of the inhibition of the immune response for the tumor to be able to grow. In another genetic screening, we perform another genetic screening. In this case, instead of patient-derived xenografts, we generated patient-derived cells, that is, cells from the pleural effusions of some of the lung cancer patients, were grown in a plate, and primary cultures were established. Again, all completely free of normal contaminant cells, and we perform whole exome sequencing and RNA sequencing of these tumors. We obtained several interesting observations that are reported in this publication of past year. But here I just want to focus in the identification of a mutation in one of the molecules that form the interferon gamma receptor, which is the interferon gamma receptor 1. One of these tumors carry, as I say, one mutation. This mutation was inactivating mutation and homozygous, supporting that this has a tumor suppressor role. When you go to the databases, you see that these mutations occur, however, in a really low percentage. We extended our analysis to a panel or a set of lung primary tumors, and we observed that these mutations are present, but the frequency is really below 1%. This is the cell line, or the primary culture that we established did not respond to interferon. You don't see an increase in phospho-STAT or in IRF1, nor in PD-L1. However, when we clone and restitute the expression of the receptor, the cells were then able to increase the levels of phospho-STAT, of IRF1, and also of PD-L1, supporting that the inactivation of this gene is also affecting the response to interferon. And here is the same. Several well-known targets of the interferon pathway were also able to be upregulated upon restitution of the function of this receptor. So if we gather all the genetics regarding genes involved in the immunoresponse, B2M-related pathway and interferon-related pathway, we can see that alterations at any of these molecules can account for about 15% of the lung tumors. The most common genes mutated are B2M and JAK2. However, other genes can also be affected. We also found that these mutations tend to be mutually exclusive. Of course, a tumor, because the effect is the same, don't need to alter more than one molecule to have the same effect. And also these mutations were present concomitantly with different well-known oncogenes and tumor suppressor genes. Here I put just MET and STK11, but the same happened with P53, with P16, with P10, and with a large number of tumors. So we think that at least two important pathways are affecting the capability of the immune system to recognize and to kill tumor cells. And one is the antigen presentation or immunorecognition, mutations at B2N mainly, probably of HLA1, HLA-A, B, or C genes. This is an issue that has been underexplored, mainly because of as I told you, because of the difficulties of genetically scrutinizing these genes, which are genetically highly variable. And also in a lower frequency alterations at other molecules that code for, or that other genes that code for proteins involved in the maturation of the complex can also appear in cancer. And another type of function that is altered in cancer and that also affects the immunoresponse is the response to interferon. mainly mutations at JAK1, as they are the most common, and less in lung cancer, but also mutations at JAK1, at interferon gamma receptor 1, 2, etc., can also be found in human tumors, indicating that these are important pathways to be considered for understanding cancer development, but also probably could be markers that will determine the lack of response to patients to immunotherapeutics. Although more studies, genetic studies need to be taken and clinical trials need to be performed considering these elements, I think that soon we will see how the genetics of these molecules will be incorporated in our selection of patients that are candidates to be treated with immunocheckpoint inhibitors, not only the levels of PL1, which is the marker that is currently being used, but that is not a perfect marker. Here just is an overall representation of different markers that can be used for selection of patients. These are the markers that I just showed you, the genetic alterations in immunorecognition related molecules or in the molecules that are involved in the response to interferon. Other oncogenes and tumor suppressor genes are under study to see whether they can be used for selecting patients. Some of them are not clear, but I just put here this information because at some point they have been proposed for this purpose. And also, on the other hand, other markers that could be used to predict sensitivity, such as the amplification of PD-L1, among other markers that are still, there is still not conclusive results regarding its use for this purpose. And that's all. I just want to acknowledge the different funding agencies that have allowed us to perform these different projects. Also, different previous members of the group that have performed a key role in these projects, Carolina Pereira and Maria Saigi, and the current members, mainly Juanjo Alburquerque and Eva Pros, that have performed essential work related to these studies. And also, of course, different collaborators that have been extremely valuable for developing the studies. And that's all. If you have any question, I will be happy to answer to you. Thank you.
Thank you very much, Montse, for this very beautiful and very, very interesting presentation. Now we have some time for questions. I noticed that there are so far 2 questions from the audience, and we will have time for more if they arrive. Let me see. The first question is from Damiana Alvarez, and it is, I read. In the case of B2M mutation and subsequent lack of HLA complex assembly on the tumor cell surface, what happens with NK cell-mediated cytotoxicity that can be triggered by the lack of HLA expression?
Well, this is a very good question. I think a lot of people that is working in this subject, in the detection of mutations on B2M and in the lack of response to immunotherapeutics, has been thinking about that, but the truth is that at this point there is no answer for that. We don't know whether the tumor has also any other mechanisms that avoid the recognition of the action of the natural killer cells. This is possible, but I have no, and I think nobody does have a clear answer for this very important question. But the truth is that the B2M is in max. It contributes to masking the immunoresponse, and even in the case of lung cancer, B2M mutations have also been found as a mechanism for resistance to immunocheckpoint inhibitors. That is in mouse models that have been administered immunotherapeutics and that have responded very well at the beginning. At the end, they become resistant to these therapeutics by acquiring mutations in B2M. And we don't know why the natural killers don't have any action on that. And this is absolutely a key question without answer so far.
Thank you very much. There is another question from Pedro Blecua. You mentioned that MET suppresses the lung tumor immune response, and that MET correlates with increased PD-L1 expression in the tumor. If a lung cancer patient has altered MET, specifically again, how likely is the patient to respond to anti-PD-L1 inhibition? What about if we extrapolate to other solid tumors?
Now, essential question. There are several ongoing clinical trials that are evaluating that. However, here there is a question, and the question is that MET is an oncogene, is a growth factor receptor, and there are inhibitors against MET. So the therapeutics in this case, it would be a little bit complicated, because what do you do? Do you inhibit the MET, or do you use PD-L1? What would be best? Should you give the patients the two drugs simultaneously? Should you try to do the drugs sequentially? We don't know yet. Yeah, there are clinical trials ongoing in this regard, because this is an important issue to be solved, especially because, I mean, there are other treatments for those tumors that have MET activation.
Thank you very much. And we have another question from Laura Martinez Verbo. Apart from the MDS case, do you know any other genetic alteration of the ones you mentioned that appear in other hematological malignancies?
I suppose she is referring to the JAK2 activating mutation. In the case of hematological diseases, there is an activation of JAK2, which is the most common, but there is also genetic activation or genetic alterations in the calreticulin. In the calreticulin, which is a protein that is involved in the maturation of the HLA-1 complex. And it is quite well known that most tumors that don't have activation of JAK2, they do have alterations in the calreticulin gene, in the case of these hematological diseases. It is interesting because there have been also observed some solid tumors with alterations in calreticulin. But there are, in the case of solid tumors and calreticulin, the data is still very scarce. In the case of hematological diseases, myelodysplastic syndromes and others, the presence of calreticulin in JAK2 wild-type tumors is quite well established.
Okay, thank you. I have two questions, and we will let some more time for the audience to think about some questions. One is, lung cancer is far away from my expertise, so forgive me if it's a naive question. Which patients undergoing immunocheckpoint inhibitory strategies previously underwent through a previous test to know if they have a cold or hot immunophenotype?
No, to my knowledge, currently, I mean, other than in perhaps some clinical trials or some research groups, no, there is this kind of routinely, the only marker that is being used for preselecting patients is PD-L1. And not all the patients that have negative PD-L1 are discarded for these therapeutics. So there are no clearly rules for preselecting patients for PD-L1 therapeutics. This is one of the problems.
Do you think that eventually it will happen, this previous test?
Yes, I'm sure it will happen. I mean, we don't know whether the levels of HLA combined with CD8s or with other immuno-related markers, or the genetics by themselves, or complementary, or everything complementary. Yes, I think so. I think this will be required to be performed at some point. First, after good clinical trials have been performed to validate the efficacy of these biomarkers, But I think these are very, very promising biomarkers, in my impression, yes.
Thank you very much. And if I understood okay, there is a low response in general to treatment for from lung cancer patients, and I would like to know to which point some of the mutations that you described are responsible for this response.
You mean for the traditional therapeutics, for the traditional target? Well, we perform an analysis with HLA-1 and B2M, and this the levels of these transcripts, because this was an analysis from databases, didn't have any association with overall survival in treatment-naive patients, nor in patients that had been treated with traditional therapeutics. Probably this needs to be performed maybe with immunostaining, maybe with different set of patients, I don't know, but probably, I mean, I will not expect unless that there is an association between these two parameters. I may be wrong, but my guess is that, I mean, unless you are treating the patients with immunotherapeutics in general, because you might have, for example, high levels of HLA or presence of HLA complex in the cell membrane, and the same tumors might have a mutation in JAK2. So these things are not, the correlation is not completely exact between the immunostaining and the genetics of all these immuno-related markers.
Okay, thank you very much. I think that we don't have more questions. Thank you very much for your presentation. And thank you everyone for attending.
Thank you very much for everything.
Good afternoon.
Good afternoon.